Hello everybody, today we'll talk about lecture number 4 from Alternative Molecule 2, Alternative Splicing. Today's topic is Alternative Splicing, and this lecture is part of Molecular Genetics 1 MLGN 301 2006. In today's lecture we will talk about a very important technique that happened in the cell alternative splicing. This new concept that we have to understand, and we have you can see many recommended books for further reading in these topics. But in this lecture we have to know and understand what is alternative splicing and how it works, why alternative splicing is important, and the splicing mechanism and different examples of alternative splicing. So first we have to understand what is splicing. RNA splicing, of course, we know that it is a process that removes introns and join exons in the primary transcript. Of course, when we know that the intron is usually contain a clear signal for splicing, so this helps the RNA, the spliceosome, to remove this intron. So we have to find a clear signal in the beginning of the intron and in the end of the intron. And of course we have many examples, like for example here we have the beta globin gene. This alternative splicing happen in that gene. Other cases like sex lethal gene in fruit fly in Drosophila. Also the splicing signal may be masked or by a regulatory protein, resulting in alternative splicing. This is another case. In some other cases, in the rare cases like HIV virus, the genes, the primary transcript pre-messenger RNA may contain several ambiguous co-splicing signals, resulting in few alternatively spliced mRNA. So in any of these cases, like the technique, what happened in Drosophila or what happened in HIV, in either of these cases, RNA splicing also always happen in alternative ways. There is alternative ways to splice the RNA. So let's see what the meaning of splicing signal. So most introns start from the sequence GU and end with the sequence AG. So this is signal to mark the start of the intron and the end of the intron. This, of course, in the direction of 5' to 3'. They are referred to as splice donor, which is GU, and splice acceptor, which is AG, respectively. However, the sequences at the two sites are not sufficient to signal the presence of intron. So of course, this means that there is something else, a third signal, that this third signal indicate the beginning and end of these splice start and end intron. So for example, another important sequence is called branch site. This branch site located 20 to 50 bases upstream the acceptor site. So this means you have to have these three important signals. So the first one is the start intron, the start, which is GU, and the end, which is AG, and in between is the branch site. So the consensus sequence of the branch site usually is CU, and then any purines like A or G, and then A, which is conserved, and then any pyrimidine like C or U. Let's see, this is how it looks like. Over 60% of the cases, exon sequences is start like this, AG, and then this is called splice donor, and in the end we'll have the splice acceptor, which is start after AG, so like that. And of course we have the branch site. This is the branch site, which is that CU, PU, and then A, and then pyrimides, as we know, CU, and then purine, and then A, which is conserved, and then any pyrimides like C or U. And then of course we have this pyrimidine rich region that always in between the branch site and the splice acceptor. So this is the typical typical feature of intron, and that's why we can very simply can program the computer and make any bioinformatic software that can detect where the intron can be expected to be found. So because this important feature can easily be programmed in a computer to find introns, and of course can predict where is the exons and where is the introns. But anyway, so the splicing mechanism, of course, the detailed splicing mechanism is quite complex, but it involves at least 5 small nuclear RNAs and their associated proteins, which called ribonucleoproteins. These ribonucleotides from a large 60S complex called spliceosome. So the spliceosome actually contains ribonucleoproteins. Then after two-step enzymatic reaction, of course, the introns is removed and the neighboring exons are joined together. The branch point or branch site residue plays a critical role in enzymatic reaction, as we will see in the movie. After that, we will see that is very important in the enzymatic reaction. Anyway, so in this case, we can see that there is a transcript, very simple transcript, have 2 exons, exon 1 and exon 2, and the intron in the middle. So simply these units, which is these the units of protein U1, U4, U2, U6, U5, these units are gathered in the beginning and try to this is the building blocks of the spliceosome during the RNA splicing. So you find that U1, U2, U4, U5, and U6 denote the small nuclear RNA and their associated proteins. The U3, as you see, you cannot find any U3. So you can see that these are the protein without U3. So what is the U3? The U3 small nuclear RNA is not involved in the RNA splicing, but involved in the processing of the pre-ribosomal RNA. So this is very important to know that all these units gather together to come together to cut the beginning and end of the intron and remove it, but except the U3 small nuclear RNA, which is not involved in the splicing, but in the another step, which is the processing of the ribosomal RNA. Okay, so let's see here an example. Here is a pre-messenger RNA from 5' to the 3', and as you see, this is the transcript which come out of this primary transcript. So we remove these introns, as we see that the AG is the start, and the start splice donor, and here the splice acceptor, and then this is the intron which will be removed by the spliceosome. And here is another one. So simply the introns, the exons, which is in the red color, will be gathered together in one messenger RNA. Of course, in this case, we have this beta-globin gene. We have also the cap here. This pre-beta-globin gene, the methionine will be removed, and then it will be, and the valine will be only exist. This is the mature beta-globin, doesn't have methionine but only have valine in the initiation of the protein. But anyway, what I want you to focus in that in this case we have removed these introns to have by the splicing mechanism to have a primary transcript. And this primary transcript, after removing, we have a mature transcript, and the mature transcript and then transcribed in an alternative way to have this final protein. But anyway, let's see, for example, these 2 examples that give us what is the meaning of alternative splicing. The first example is a 6 lethal gene. The sexual differentiation in Drosophila fruit fly is regulated by a protein called 6-lethal SXL protein. The female embryo express functional SXL proteins, whereas the male embryo express non-functional SXL proteins. Okay, so these are the differences. So SXL protein exists only in the female, and it's functional in the female, but in the male it's not functional, and we will see why. The differences, the main difference is not from alternative splicing, as shown in the following figure. Let's see the following figure and then concentrate on what happened. In this example, we have first of all, this gene have alternative promoters. So this alternative promoter we have already mentioned in the last lecture, that these two promoters are controlling the same gene, one for the early stage and another one for the late stage development. So this promoter for the early stage development of this protein, and this for the late stage development of the embryo. Okay, in the female, the early stage will of course come because it's early, so this promoter will work early. So this promoter will help the transcription of the first messenger RNA of this gene, and then we'll have in that gene we will have one intron and two exons. So in these two exons, number 1 and number 2, we'll have a normal splicing and remove this intron, and then we'll have early female SXL protein. So the resulting from the first and early stage splicing, that we have two exons, exon 1 and exon 2, and remove the intron and have early female SXL protein. This is in the early stage. In the late stage, the promoter number 2, which is called PL, the late stage promoter, will work in this case, and then the production will from the primary transcript will have four exons, 1, 2, 3, 4, 1, 2, 3, 4. Okay, what happened is that the early female protein, which is already exists from the early stage, will mask the signal of the third exon, will mask it. So in this case, the cutting enzyme, the spliceosome, will skip the removal of this exon, of this intron. So in this case, we'll find that he will remove the exon number 3 with the introns. So in this case, we will find that the resulting transcript will be having 1, 2, and 4. 1, 2, and 4, and this is a functional protein called late female SXL protein. Again, so we have in SXL protein in the female happens in 2 steps. In the first steps, in the early stage, the early stage promoter will work, and the protein will work with 2 exon only and 1 intron. This intron will be removed and have a non-functional early female SXL protein. In the late stage, the same gene will work with another promoter called PL promoter, which is late stage promoter for late stage. In this case, when we use this promoter, this will result in 4 exon. The existence in the transcript will have 4 exon. And exon number 3 in this case will be masked. The signal to remove this intron will be masked using the early female SXL protein, which have been produced in the early stage. And then in this case, the final protein will contain 3 different exon, which is number 1, number 2, number 4. And this protein, which have 1, 2, and 4, is functional, but the first one is not functional. Okay, Let's see in the male embryo. This male embryo, in the male embryo, only the late promoter will work and produce, as we see, number 1, 2, 3, 4. And of course, because in the male embryo doesn't have the protein in the early stage protein, so in this case the 4 exon will stay intact. In this case, produce a protein which contains this 4 exon is non-functional male SXL protein. So only the female protein will be functional, and but the male SXL protein will be not functional. In this case, we have only this produce males. So here we can have the description, expression of the receptor sex lethal SXL protein. In the early stage, as we see, of embryogenesis, the SXL protein is expressed in female embryo, but not in the male embryo. In the late embryo, in the late female embryo, the SXL protein produced in early stage may mask the splicing signal for the second intron, resulting in a different protein than in the male embryo. The gene which encodes the SXL protein contains 2 promoters denoted PL and PE. PL for early promoter and PE for late PL for late promoter and PE for the early promoter. PL is active in the late development of both female and male embryos, but PE is active only, and this is only difference, in the early stage of the female embryos. Therefore, in early embryogenesis, the XL protein is expressed only in the female embryo. The primary transcript generated by PL consists of 4 exons separated by 3 introns. In the male embryo, the 3 introns are removed and all the 4 exons are joined together. Its product is a non-functional protein. But in the female embryo, the SXL protein produced at the early stage may bind to the spliced acceptor site of the second intron. As a result, the splicing machinery takes the next acceptor site for splicing. The third exon is then skipped, producing a functional SXL protein. Exon skipping is also frequently observed when a critical residue is splicing signal is mutated. So this technique, the one that we have just mentioned in the Drosophila, is called exon skipping. So as we see that because of the early stage production of that protein, this masks the splicing signal, and that we call it exon skipping. There is another example which happened in rare species like HIV. For example, in the genome, the HIV genome contains 9 genes: GAG, POL, VIF, and VBR, and VBU, and envelope ENV, and NEF, REV, and TAT. Anyway, these nine genes, their proteins products are all derived from a single primary transcript. And of course, this happened because 3 different mechanisms. One of them are alternative splicing. The second 2 we'll talk about them later, but today we'll talk about alternative splicing. The second 2 are leak scanning of initiation codon and ribosomal frame shifting. But one of them is alternative splicing. Is very important to have 9 genes produced and 9 proteins produced from single primary transcript. Here we can take a look and see that one single primary transcript in HIV can produce 9 different proteins, and this is one of the marvelous example we can find in nature, that how small piece of messenger RNA can result in all these different proteins. Okay, why alternative splicing is important? Alternative RNA splicing is very important for the maintenance of gene function and regulation of expression of certain genes. For example, the same primary transcript the same messenger RNA, for example, can be spliced variously in different tissues, cell types, and in different developmental stages, and react to various biological signals. So far in human genome, only, of course, we found about 22,000 genome or something have been so far identified, but compared with previous estimate of 100,000 or more. This indicates that one gene encodes more than one distinct messenger RNA. Hence, more than one protein may play a critical role in expanding the function of our genomes. What I want to say in that's why we have a very limited number of genes in our genomes or in eukaryotic genomes. However, we can find a larger amount of proteins that can be produced, and we thought before that we have a much more number of genes, but actually we have a limited number of genes produce a large number of proteins. As we see that one primary transcript can result in many alternative proteins and many different proteins because of alternative splicing. Of course, in nature we have many examples. We just mentioned one or two, but in nature you have many of them, and there is actually a database that we will see in the end that can predict and can this database can have a lot of examples of newly discovered gene with alternative splicing mechanism. Of course, as we see, is a very important terms. There is many important terms in alternative splicing, like lariat, trans-splicing, and different other terms that people may use when we talk about alternative splicing. And as we said, that there is two ways in alternative splicing, and we have mentioned one of them that happened in Drosophila and another one in HIV. As we see that alternative splicing can be either constitutive or regulated, and this is two different ways of alternative splicing. But in all, that there is online database for alternative splicing, as we mentioned before, and this database contain actually a lot of examples of alternative splicing, and any newly discovered organism or gene that have been identified with alternative splicing, just deposit in that database. Okay, the practical questions in this session is: what is alternative splicing? Why is alternative splicing important? Describe two examples of alternative splicing. What are the differences between constitutive and regulated alternative splicing? Thank you for listening, and I hope you can join the channel and learn more about other topics in molecular genetics and bioinformatics and genomics. Thank you very much for listening, and hope to see you again.